these tRNA need to, you need to find the complementary code to figure out what the amino acids that's attached to them are. So to find the complementary code, you take the letter and you find what matches with it. So U would be matched with A would be matched with U, C would be matched with G, and G would be matched with C. To figure out which amino acid is needed, you can look at one of these charts. So this one is CCG, so it would be GGC. So you go from G to G to C, which would be that, which is glycine. So for this one, UCA would be AGU. So AGU, which would be this, which would be serine. Then for this one, CAG would be GUC, GUC, which is this, AUG, which is this, which is methionine, which is also the codon for starting the whole process. So to make RNA, the DNA, RNA polymerase pulls the DNA apart, like that, and then it finds the complementary code and makes that into a strand of RNA. But as you can see, there's no thymine in the RNA because RNA has uracil instead of thymine, but it still binds with adenine. After the mRNA is made, because this is messenger RNA, it leaves the nucleus through the nuclear pores and moves over into the cytoplasm. This is because the DNA can't leave the nucleus.
After the mRNA goes through transcription, it binds with the small ribosomal subunit and the large ribosomal subunit. Basically, this is the start of translation. Like before, a tRNA basically comes down and methionine usually kind of is the first one, and it starts in the P site. And then another one, GGC, G, comes in at the A site. And basically, as they switch sites, methionine and lysine form a polypeptide bond. So tRNA is basically an anticodon, obviously tRNA, and then an amino acid. And the anticodon is really super important because that is the thing that binds with the mRNA, which gets basically the amino acids to bond, which then makes the proteins, and proteins are super important in a cell.